Hello and welcome to my next live stream. It is the A2 Unit 4 live stream. So is my sound quality and my video quality and all the qualities and levels fine for everyone? And if so, we will start. It's weird, I've only got 2 viewers considering about an hour ago, an hour before the live stream started, I had about 5 or 6. Bit weird. Anyway, right. Start. First question I've been asked is oxidative phosphorylation and cameo osmosis. Now, first thing different between them, there really isn't. Oxidative phosphorylation is the whole process along the mitochondrial inner membrane by which ATP is produced. So from electrons in the carrier proteins, which we'll go through, to actual ATP being produced. Chemiosmosis is specifically the bit where hydrogen ions go through the ATP synthase to produce ATP, but we will go through that. So, first things first, I'm going to draw. Right, so this is your inner mitochondrial membrane. That's the inner so I'll put inner membrane, outer membrane, intermembrane. So inner membrane, outer membrane, and that's the intermembrane space. Now, what happens is we get our molecules of reduced NAD and reduced FAD coming from the Krebs cycle, glycolysis, and the link reaction. Now, what happens here is that NAD, so we're going to go for NAD, gets oxidized. So we've got NADH2, which reduces NAD, becomes NAD plus H2. Don't worry, I will go through those questions. Yeah, I will. Right. Then the hydrogen, I'm going to draw it here. So that's the process. Hydrogen breaks into a proton and an electron. And this happens around here, around these 4 proteins, which are carrier proteins. Now, carrier protein 1, 3, and 4. Little numbers. 1, 2, 3, 4. 1, 3, and 4 will work with NAD, and carrier protein number 2 works with FAD. And also, FAD, just to mention, protons will not go through FAD, but it will not go through carrier protein 2, but it will go through carrier protein 1, 3, and 4. So we've got our protons and electrons. Now the electrons move. It's 2e minus because it's 2H, so 2e minus. Go into that carrier protein, then that one, then that one. And this is the electron transport chain. So wherever the electrons, we just show it as starting at the first electron carrier, but equally at FAD, 2 electrons could be released, or NAD here. I'll show as I've drawn across the page. Oops. Or NAD here. So it could be anywhere along the chain, but it will continue. The protons that are released will go through the carrier proteins, so 1, 3, and 4, into the intermembrane space. Then the protons will create a concentration gradient, or a electrochemical gradient, or a pH gradient, whatever you want to call it. But it's a concentration gradient, which will cause them to diffuse through this molecule, which is ATP synthase. Then, as H plus goes through here, we'll go a little bit more what actually happens here. It causes ADP. So we get ADP plus inorganic phosphate becomes ATP. Now this stage here, I will use my red pen. That stage there is chemiosmosis. This whole thing is oxidative phosphorylation. That bit there is chemiosmosis. So yeah, H plus moves through ATP synthase, and what it does, it gives energy. That's what it does. The movement of the protons through ATP synthase give energy to ADP plus PI, which can cause them to create that phosphate bond, so you get ATP. Now, at this side, we get H plus plus E minus. The E minus comes from here, forming H, 1 hydrogen. This happens twice. Remember, 2 electrons and 2 protons. So we get that times 2. So we now have 2H. And this combines now with the final electron acceptor, which is oxygen, to form water. In reality, you get 4H plus O2 forms 2H2O, but it can be shown this way as well. So let me just do it around like that. So protons diffuse down the concentration gradient through ATP synthase, giving energy to ADP plus PI, which causes them to create a phosphate bond, creating ATP. When the protons get through, they join with electrons from the electron transport chain to form hydrogen, which combines with oxygen to form water. Yep. So that's and then just remember number 1, 3, 4 NAD at 2 FAD. Does that answer the 2 people who wanted to know about oxidative phosphorylation and chemiosmosis? Or do you want any extra things to be asked? FAD is done at the second electron carrier. And once at FAD, it does the same thing as NAD, so it releases 2 hydrogens. The electrons will go into Fe and move on, but the 2 H's, the H plus, will then just go through a different electron carrier, so number 1 or number 3. So what do you mean, did you Fe2 was to do with FAD? Yes, FADs carry it carries on because the electrons can move through. It's just protons cannot go through the second electron carrier. Students, I said, oxidative phosphorylation, the whole step here, all this. Cameo osmosis is this bit here, so the actual movement of protons through ATP synthase to produce ATP. Yes, there are always 4 electron carriers. Okay, good, Alice. I'm glad I did. Hello. Right, I'm going to move on now. Next question was right to the bottom. Can I go over the loop of Henle, the step-by-step process? Yes, I can. Right. Loop of Henle. Okay. Students, yes, you're right there. Glitter, I definitely think the liver will come up. That hasn't come up for ages. CGP is 3, is there? One sec, Lauren. Let me just have a look at that. Yeah, I see what you mean. There are only 3 in the... One second. Right, well, what I'm going to say is not 100% sure, but since the OCR book says there are 4, we must say there are 4. So ignore the CGP book in this case. It is 4, not 3. Right, loop of Henle. We have descending limb, ascending limb, collecting duct. Right. So I'm right. When I say ions, I'm going to just say I. So I equals ions equals minus, and W equals H2O. Just to make it look a little bit easier. Right. W equals water, I equals ions, which are sodium and chloride ions. Yeah. I don't think it matters. As I say, since the official OCR textbook says 4, say 4, because that's what the official textbook says. Right. So we start off. We get a kind of happening. There's a high concentration of sodium and chloride ions. So high concentration I outside, low, and which means water will diffuse out by osmosis because there's low water potential outside. But equally, since there's a low concentration of ions inside, the ions will diffuse into the loop of Henle. So in the first step, water diffuses out because there's a low water potential outside the loop of Henle. Ions are fusing because there's a low concentration of ions in. And at the same time, as you go through you get water moving out of the limb. loop of Henle. But once you got to the bottom, there's now a high concentration of ions inside the loop of Henle, and there is a low concentration outside. So we get ions diffusing out again. And this is diffusion. Now I'm going to change my pen. I'm glitter girly, yes. I think I'm doing a little bit on the liver later. Right. Then in this little bulgy bit, since the concentration of ions has now increased outside the loop of Henle, there is a diffusion gradient which would make it want to diffuse in, but it doesn't. It continues going out by active transport. So at the bottom, it goes out by diffusion. At the top, it goes out by active transport. And along this whole area here, that's distal convoluted tubule and the ascending limb, it is waterproof. Water cannot pass in or out. But then, once we get to the collecting duct, there's now a very high concentration of ions outside the loop of Henle, so a very low water potential. So along the whole collecting duct, water will diffuse out. So whoever asked that, does that make sense? Right, great, good. Next thing I'll answer. Right, right. Can you guys know how to investigate rate of respiration? I'm going to do that in one go with photosynthesis a little bit later, but I'm now going to do the question 3C from the paper. Right. The question, for those who can't see it, is we're showing a diagram that represents light harvesting found in the surface of the internal membrane of a chloroplast. Basically, this sort of diagram. It doesn't look like this in the picture, but this is my way of drawing the diagram. Right. So basically, we have light entering, bouncing around randomly, and going to the photosystem. The way you want to answer this is, right, photons and their energy are absorbed by accessory pigment molecules. This excites the electrons to a higher energy level, and as this happens, energy is passed from one pigment to another randomly. Eventually, the light energy will be passed to the reaction center, which contains chlorophyll A. Chlorophyll A. The range of accessory pigments means we get a large range of light being absorbed. That's how you would answer it. It's reasonably simple. Basically, photon energy absorbed excites electrons to a higher energy level, randomly move between accessory pigment molecules, and eventually reach the reaction center, which contains chlorophyll A. Oh God, whoa. What's countercurrent multiplier? Let me go back. That answers. I hope it answers your question about the that question. Right, the countercurrent multiplier rubbish. I know it's badly explained in all the books. Yeah, it is essentially countercurrent means that there is liquid flowing one way and the other way. It's, yeah, and that's kind of right. Right, one sec. Basically, the countercurrent Ron multiply means, I'm going to draw it just the other side. Essentially, the loop Henle would actually look something like this. And here, this is a hairpin, and it's very close together, which means you get one liquid going down that way, one liquid going up that way. And because they're very close together and they're going in opposite directions, yeah, glitter goes basically answered there. But yeah, since they're one's flowing one way, one's flowing the other way, it means that you can get that transfer of ions very quickly, which creates, so you can flow out of one straight into the other, and likewise, basically flow, can diffuse into this one quite easily, and it means you get a very high concentration gradient, which is what you want. So basically, they flow in opposite directions very near each other and create a very high concentration gradient. Right. Next. Right. How the structure of a chloroplast helps it to carry out its function. Right. Before I do that, is everyone happy with everything I've done so far? Jesus Christ, yeah, it's never been done in any book I've seen, any OCR endorsed book. Yeah, Alice, yeah. Right. Good. Right. So we're now how a chloroplast is. All right. Let's draw a chloroplast. Right. This is a really badly drawn chloroplast. Right. And yeah. Right. A few things. Starch granule. As a starch granule. All right. Now I always need to check this to get this right because it's so easy to get it wrong. Right. A singular one of these is a thylakoid. A stack is a granum, and one of these is an intergranal. Let me just check how I'm spelling that. Granule, yeah. Intergranal lamellae. Where are we? Thank you. Right, so it's yeah, we have thylakoid is one membrane, granum is a stack, intergranal lamellae is a bit between, starch, and we have outer membrane, inner membrane, envelope. It's the whole thing. Now, the first thing before I go into anything is that any question ever on anything to do with biology that ever asks you to describe how something is adapted for its role, an organelle in particular, or an organ or anything, always answer large surface area to volume ratio. And here you can see here many stacks of membrane folded over each other, because it doesn't look like that. It more looks kind of like, yeah, there's kind of loads of stacks, and that creates a large surface area. That's always one. So the inner membrane has transport protein, so this will be a transporter protein. I can control what enters and leaves the chloroplast, just like any normal membrane. Right, the many grana with thylakoid membranes provide a large surface area. Photosystems are found in the thylakoid membrane, which provide a place to harvest light. There are enzymes. All the enzymes needed are found in the stroma, which I didn't label. Sorry. Like cytoplasms in a cell, stroma is the center of the chloroplast, and that contains all the enzymes needed. So you have ribulose bisphosphate, glycerate 3-phosphate, triose phosphate. All those enzymes or protein molecules which are needed are found in the stroma. It's already there. Rubisco, that's another one. I mean, so the ones I named before proteins, but yeah, there are proteins and enzymes. So they will catalyze the reactions of light dependent and independent stage. The stroma surrounds the granulum so that the products from the light dependent stage can go immediately to the area of the light independent stage. And proteins embedded in the grana hold the photosystems in place. So there are proteins embedded in these membranes to stop the granum kind of losing shape. I hope that answers your question. All right, what's the next question? Next question is just respiratory substrates. Right. Ooh. Can you talk about how photosystems have a range of pigments to absorb different waves? Right, firstly, stroma, it doesn't basically allow it to happen quickly, but it allows the time between the light independent stage, no, the light dependent stage and light independent stage from happening to be very quick. So, like, imagine if you were, building a car, and you built like the chassis and the frame of the car, and you had to then take it all the way to another country to get the rest built. It's going to take quite a long time. If you just go to like another room in the factory, it's going to be very quick. And that's what it is. Basically, since the next stage happens right in the chloroplast itself, it is very quick from one stage to the next. Can you talk about how photosystems? Let me have a look. Does it say that? I don't know. Can you talk about how photosystems have a range of pigments? Yeah, I think you could. That is definitely a function. Yeah, definitely. I say, Glitz Girl, you can definitely say that. And if they say you can't say that, then I take that back. But I assume you. I think it'd be fine to say that personally. Right. Respiratory substrates is the next question. Right. You just need to know that a respiratory substrate is just an organic substance that can be used for respiration, and that is proteins, carbohydrates, and lipids. Those are the three big ones. Right. Carbohydrates, we know we have glucose, and glucose is just naturally respired. But interestingly, carbohydrates, the one we actually respire, gives the least amount of energy when you do respire it. Proteins can be deaminated. They will be deaminated, removing the amine group, and it creates a keto acid, which can then be converted into glycogen or fat. And then if it's glycogen, obviously it can be broken down into glucose and then respired normally. If it's a fat, as we'll see with lipids, it can be converted to pyruvate or acetate and enter the Krebs. So basically, it can be entered the Krebs cycle and be respired that way. Yes, I do control heart rate. Do I have video? Yes, I do. All my videos on this unit have been completed. All my biology videos are done apart from 3, and that's in the final module of A2. So the one on responding to the environments as plants, the brain and responding to the environment as humans, and behavior. Those are the 3 videos I've got left to do. Well, honestly, I don't 100% know, but one big reason is that lipids and proteins, they both enter the Krebs cycle. Yeah, so they're Alice. Yeah, so they have to enter the Krebs cycle. The Krebs cycle is aerobic respiration, so there has to be oxygen, which means when we use glucose, we do not always get enough oxygen to respire all our glucose when we need the energy requirements. I mean, even if we are breathing, let's say if you sprint, you'll get that stitch because you're respiring anaerobically because you're not getting enough oxygen, which means that if we only respired lipids and proteins, we would have to respire aerobically. So whenever we didn't get enough oxygen, we wouldn't be able to we wouldn't be able to keep up with our energy supplies. So I think that's probably the reason. Why is the respiration equation oversimplified? No, I wouldn't say. Well, personally, I wouldn't say that's the answer. Is that question actually written anywhere? Can I have a look at it? Because I very much say it's oversimplified because that implies that it's just a one-step reaction when it very clearly isn't. It's multi-steps in that reaction. I mean, you have just all the steps in link, all the steps in Krebs, and oxidative phosphorylation. So it isn't just that exact reaction, because if you actually had reaction. All right, let's say it was that exact reaction. Now, if you remember, photosynthesis is the exact reaction but the other way around. So it's water plus carbon dioxide becomes oxygen plus glucose. Well, that would imply, if this was exactly what was happening, it would imply that they could go either way, which means we could photosynthesize because the second we got carbon dioxide and water, we could just convert it back into the other stuff. And plants, equally, the second they got any glucose, they could then just instantly respire it. And then the second they respire, they could turn back into glucose, and makes the whole kind of thing weird and not pointless. So, yeah. Yeah, that's another reason. So, yeah. Ellie, that's another reason. Yes, there are many reasons why we use glucose. Glucose is a lot easier to store than lipids. That's definitely true. But I mean, proteins are quite easy to store, and I mean we use them naturally. Yeah, yeah, reduces. I think about using glucose, something like 70% of the energy um, found in glucose is converted to heat. A lot of energy. It's yeah, everything's just converted to heat. um, I guess that could very well be true. But yeah, I hope that answers your question. Right. Right. Next question. I saw one about. Right. Someone asked about ADH. Can you be a little bit more? What do you mean by A? Do you mean like osmoregulation or what actually ADH is? There's one on pregnancy testing. Here we go. Pregnancy testing. Right. There's a hormone. I need to human chorionic gonadotrophin. That is a hormone you need to remember. Now, we'll call human chorionic gonadotrophin, HCG, we'll call it that. And basically, it's found when a woman is pregnant. Now, on the pregnancy test, you will have antibodies which are complementary to HCG, which means when HCG has gone on the, when a woman pees on the stick and it has HCG, it'll bind to these antibodies. And these antibody complexes will then move up the strip until it sticks to a band of immobilized antibodies. And these antibodies carry a blue bead. So essentially what you do is you just get a blue line moving up a pregnancy test. So HCG is complementary to antibodies, binds to the antibodies which carry a blue bead. This causes them to move up the stick until they reach another blue line. So you get 2 blue lines. And you have one permanent blue line and one that will move up. The one that moves up is basically, you then check that with the one that's there. Yeah, I hope that answers your question. Osmoregulation, okay. Right. Well, I'll do co-transporter proteins and selective reabsorption, and then I will do osmoregulation. Those two go quite nicely together. Right. I'm going to draw it really badly. Right, this is the blood. This is a cell of the proximal convoluted tubule. Right, it's not, not amazingly well drawn. Right, first things, sodium ions are actively transported out of the proximal convoluted tubule no, I can't draw a picture of a pregnancy hormone thing. I'm not that skilled in art. Anyway, sodium ions are actually transported out of the proximal convoluted tubule cells into the blood. Now this means here, sodium ions then diffuse into the cell down their concentration gradient. So you have high concentration here. They're actively transported out, lowering the concentration here, causing it to diffuse in. But they diffuse through co-transporter proteins, and so they bring with them a little bond, either glucose or amino acids. And so you have sodium ions, glucose, amino acids, and then glucose and amino acids will diffuse into the blood. So sodium ions actually transported out. Sodium ions diffuse back in through co-transporter proteins, bringing glucose or amino acids with them, and the glucose and amino acids diffuse into the blood. Okay, thanks for answering that question. Right. Interesting fact, actually, if a man urinates onto a pregnancy test and he gets pregnant, it's a early sign of prostate cancer. So men can actually get a positive pregnancy test. Very quick, I'll just do King Genetics question because it's very easy. It was actually done on the most recent paper, June 2014, June 2013 F214 question. The first question showed it, and it's like, right. So if that's from face on, if that is the neurone, the way it's wrapped round is. One sec. Let me just. I will admit it is not the easiest thing in the world to picture. I really hope there's a little diagram in the book for me. Ah, there is. Here we go. Right. You have the neuron, and it literally is just. You get that wrapped round, and eventually you'll have like an end. You have a nucleus. Let me just. There you go. So there's literally just a neuron with a cell wrapped around it, like almost like a snake wrapped around a branch, which actually does look quite like. Little circle is the axon. Well, not necessarily axon. It could be a dendron. Whatever the part of the neuron is. So I think they can be attached to dendrons as well, can't they? Yeah, of course they can be attached to dendrons. Yeah. Because it could be a dendron or an axon. Yeah. Analysis like that spiral. This is the neuron. But it's a Schwann. Again, it doesn't have to be an axon. It could be a dendron. But don't worry, you won't need to know this for the exam because this was literally on the last paper where they showed a picture of it and asked you to talk about myelination. Yeah, it is. Actually, it's, there you go, evil snake. Because I'm weird like that, and decided to draw an evil snake. Now, does that answer your question? Whoever asked about it. Myelin is the kind of, I believe it's the kind of cell membrane of the Schwann cell. I believe that's what myelin is. So it's part of the cell. Yeah, sure. Do control of heart rate. I believe Glitter Girly kind of explained polyclonal. I actually can say now she explained that a lot better than I could have because I've actually. I'm going to be completely honest. I don't know what that is. I've never come across it, polyclonal and mono. So if someone else can explain that, I literally don't know. Oh, obviously, he does produce it then, doesn't? Yeah, I thought they didn't produce myelin. I thought they kind of. Yeah, I don't think they produce myelin. They're made of it, unless of course since the cell itself kind of makes bits of the cell. What do you mean when it says one Schwann cell is around many neurons in unmyelinated neuron? Because it won't be. You don't have Schwann cells in unmyelinated neurons. Can you explain that, King? I'll quickly do heart rate, control of heart rate. Right. Now this is something else which isn't awfully well done in the book. Right. Oh, I remember I didn't like that book, did I? I don't like the way the textbook does it, so I use a different book for control of heart rate. There we go. Right. If my because some neurons don't need to be. You don't need to have as quick a reaction. Let's say that really two neurons really close to each other. You don't need it. Yeah, I'll do more osmo. I did promise the osmoregulation. Yeah. Right. So you have a brain. You have a heart. Right. Accurate drawings there. Right. There are 4 conditions you need to know about when it comes to heart rate, and that is how heart rate is changed in response to high blood pressure, low blood pressure, and high pH levels and low pH levels. So I'm going to do a little here. Pressure is detected by baroreceptors. pH is detected by chemioreceptors. So baroreceptors will detect a change in pressure. Now, I'm going to do another. Right, you'll see what I've just done high and low. In response to high, well, firstly, in response to anything, it'll go along sensory neurons. So this is going to be sensory to the brain. Then, when it's high, it's parasympathetic neurons. When it's low, right, sympathetic. Sorry for the awful spelling there. Parasympathetic. And then when it's low, it's noradrenaline. And that's acetylcholine. Acetylcholine. I'll explain that now. Right. Yeah. So when it's in response to something, when something's too high, the brain will send a signal down the parasympathetic neuron, and it will release acetylcholine and have an effect. When something is low, it'll go along the sympathetic neuron and release noradrenaline. And this applies to both pressure and pH. So now I will actually kind of explain. Yes, yeah, it is CO2 and O2 as well. I'm just doing pH just for simplicities. I don't worry, I'll go on it at the end. But yeah, just I didn't want to write say PHO2 and CO2 all the time, but don't worry. Basically, so let's say there's high pressure. So this is what you do: high pressure. It'll be detected by the baroreceptors, go along a sensory neuron to the brain, which will send a signal down the parasympathetic neuron and release acetylcholine. If the pH CO2 or O2 is too high, detected by chemioreceptors, signal sent along sensory neuron to the brain, too high, so down the parasympathetic and releases acetylcholine. That's if it's too high. Pressure, if it's too low, detected by the baroreceptors, sensory neuron to the brain, then if it's low, goes down the sympathetic neuron and releases noradrenaline. If pH CO2 or O2 is too low, detected by chemioreceptors, go along sensory neuron to the brain, then down sympathetic neuron and releases noradrenaline. So if it is ever high, it's always down. It's Vegas and accelerator. One second. Oh, I've got to say, in the brain, the section is the medulla oblongata. I've got to say that. Right. Okay, one sec. Ooh. Yeah. Yeah. Basically, acetylcholine will cause, will cause the vagus nerve to kind of lower the heart rate. But right, the chemo and baroreceptors are found in the blood vessels near the heart, so like I think the aorta. No, that book, as usual, is useless. Yeah. Yeah, baroreceptors and chemioreceptors are found in the aorta and the carotid artery. They're found in in the blood vessels near the heart. No. The OCR spread is awful. What does do it well is this book. This double spread here. Sorry, I'm not working out this camera. It explains it very well here. But yeah. Now the way you need to remember this is that, let's say your blood pressure or pH levels are low, particularly blood pressure, so you're kind of a bit low. This is a really weird way of thinking about it, but let's say your body's a bit tired, a bit low on energy. Your body will be sympathetic to you. It's understanding that it's understanding that you're very tired. It's being sympathetic, so it's going to give you noradrenaline, which is very similar to adrenaline, to kind of make you a little bit happier, give you more energy. So if you're tired, body is sympathetic to you and give you some adrenaline, or rather noradrenaline, to help you out. And just remember the opposite for if it's high, it's parasympathetic, it's not being sympathetic to you, and it releases the other molecule, acetylcholine. That's the way I remember it. King Genetics, I don't know what will come up on these year's paper for the A2. I think the liver will. The liver will. A hard question on photosynthesis and an easy question on respiration, probably, and one on experiments, and then something on hormones rather than the nervous system. That's what I think will come up. Yeah. Does that make it clear for everyone about control of the heart rate, or do you want me to go over it again? Yeah, low anything, low of either of these two will cause the heart rate to increase. That's quite a good way, yeah. Yeah, if it's low, anything's low, heart rate's increased. If anything's high, heart rate is decreased. I I promise osmoregulation, then liver. Um, don't worry, osmoregulation really isn't that won't take that long at all. Osmoregulation, right? Basically, all right. Right. Now, just so you know, guys, the order I'm doing it in, I'm going to do the osmoregulation because I did promise that next. Then the liver, I will do that afterwards, and then I'll do photosynthesis practicals. But really, you do not need to know them. They will never ask you those ones. Right. Osmoregulation. So we have antidiuretic hormone, which is released from the pituitary gland in the hypothalamus or near the hypothalamus, I believe. I don't. Yeah, it's near the hypothalamus, and from the posterior lobe of the pituitary gland. So osmoreceptors are found in the hypothalamus. These detect changes in water potential, and what they do is they respond um, by stimulating neurosecretory cells in the hypothalamus, which will produce ADH from the posterior pituitary gland. So osmoregulators in the hypothalamus detect the change, causing ADH to be released from the posterior pituitary gland. Now, we're gonna assume, just for the way I explained now, that there's a water potential is lower. There's a decrease in water potential than what you'd expect. So that means you haven't got as much water in your blood. This will cause more ADH to be released. It goes to the collecting duct. This is the collecting duct, and we're going to zoom in there. That's a cell. Nucleus. Um, so ADH will go to the kidneys, to the collecting duct, and bind to receptors. So I'm have literally. That is my receptor, and it binds to ADH. The molecule does not look like an ADH, but I've just drawn it like that. So ADH will bind to the receptors. This will cause a series of enzyme-controlled reactions, which causes vesicles containing water-permeable channels called aquaporins to fuse to the membrane. Here, so we get water channels. These water channels allow more water to be sent out. So if more ADH will cause more aquaporins, more water channels, which means more water can be reabsorbed, so the water potential of the blood will increase. If the water potential of the blood is too high, less ADH is produced, so less ADH will bind to receptors, so less water channels are made, so less water is reabsorbed. That's osmoregulation. Does that answer your question? Oh, and also ADH is slowly broken down after a while. It has a half-life of about 20 minutes, and half-life means that within 20 minutes, half of the concentration of ADH will have been broken down. Does that? I hope that answers all your question there. Great. Now, as promised, the liver, structure of the liver. The structure of the liver is pretty simple. You have four main vessels. You have hepatic arteries, hepatic veins, hepatoportal veins, and the bile. And oh, I don't know how to say this. Where is it? Bile. It's a bile canaliculus. Yeah, canaliculus. I have no idea if that's how you say it, but that's how I'm going to say it. Um, Hepatic artery takes oxygenated blood from the heart to the liver. The hepatic vein takes blood from the liver, that's deoxygenated now, to all the way around again back to the heart. Hepatic portal vein takes all the contents that's been digested from the stomach and in the digestive system into the liver, and the bile canalicus creates bile and transports bile to the bile duct to be stored. So hepatic artery, because hepat, that is kind of the I believe it's Greek or Latin, one of the two, for liver. So we have hepatocytes and liver cells, and they hepatic artery oxygenated blood to the liver, hepatic vein deoxygenated blood away from the liver, hepatic portal vein is the stuff from the intestines and digestive system into the liver, so it can be deaminated or detoxified, and then bile canaliculus takes bile created by the liver to the bile duct. And the way this is arranged, we have, I'll draw this really badly, hepatic vein. We then have, I say this is literally going to be the worst drawing ever, but hepatic artery, hepatic portal vein. These go weird legs, but yeah. So the hepatic artery and the hepatic portal vein join together to form something called a sinusoid. Should be here. Sinusoid. And this will run all the way to the hepatic vein. You have many of these all around in a lobule, which is like one section of the liver. So you could have many livers around the hepatic vein, and that's a lobule. I'm drawing it, don't worry. I'll go over Kupfer cells. You also then have the bile canaliculus, which doesn't quite join in with anything else. But this will then be connected to the gallbladder from the bile duct, will take all that away. And all the cells between, those are hepatocytes, liver cells, and they will produce all the chemicals. They'll produce bile, and they will take in the contents from the hepatic portal vein to be detoxified and deaminated. And then in this you'll have Kupfer cells, which are types of white blood cells. They are macrophages. They move about within the sinusoid and break down and recycle old red blood cells and any other diseases which just happen to be around in the liver. And they also, since they break down red blood cells, they break down hemoglobin, and that produces bilirubin, which is basically, I think it's the brown pigment in the fecal matter. So does that answer your question? I don't know what drawn dice is actually. Sorry. Oh no, that's just right. So the branches of the hepatic vein, so more branches, just show that it's not just here. It's not just this bit here. There are loads of different lobules all around the hepatic vein. So it has many different sinusoids joining to it. Um, no, the bile canalicus collects bile from the liver cells. Question with the photosynthesis practicals. Honestly, I'm not 100% sure what you want me to go through on this because it's essentially just learning a practical, which I would just have to read out. Um, can you tell me exactly what you don't get about this? But honestly, I can't see this really being questioned upon. They're not going to ask you this exact practical. I wouldn't. I doubt it. So can you say what exactly you don't understand about it? Because if it's just you don't. Um, oh, did I say creates it? I didn't mean to. I'm dreadfully sorry. I said creates it. It just carries. The liver cells create it, and it carries, and the bile canalicus carries the bile. Sorry about that. But yeah, can you say what you don't understand about the practical? Because if it is actually just you don't know the practical itself, I'm not saying that's what it is. Just I'm saying if it is that, I'd rather not go over that purely because that's just me reading off a textbook, which, as has been voiced in previous live streams, that it was a terrible bad thing for me to do to read from the textbook. So I won't. Again, person who asked about dialysis, I think. What specifically about dialysis do you want me to go over? Because again, dialysis is just kind of learning from the textbook. Um, like, do you want me to go over advantages and disadvantages of dialysis, or one specific form of dialysis, or just yeah? Whenever asking, try and be as specific as possible. Helps me a little bit. The practicals, not really. No. I would just read over them so you have a base knowledge, but the questions they ask on the practicals, they're never going to ask you to write down the method of that practical because it'd be like writing an essay on it. They'll never ask you to write down the practical. So if you ask you questions on it, and it's common sense, so you no learning that practical off by heart would be a little bit pointless. And generally, most things on how science works is a little bit pointless pages. But read it, go over it. But I doubt it's going to come up really. There's an outline of both dialysis, right? So we have hemodialysis and peritoneal dialysis. Peritoneal. So that's probably it. Peritoneal. I'm always sorry if I ever pronounce any of these words wrong, but everyone knows with biology, if there was an oral speaking test, everyone would fail because it's biology is full of complicated words that no one can pronounce. Uh, yeah, that's pretty much it. Coenzymes, what? What? Coenzymes question mark, what? Right. Hemodialysis is right. Basically, your blood is removed from your arm. Um, you have the chemical heparin is added to avoid blood clotting. Any air bubbles are removed before the body, so it has like an air trap to remove air bubbles. If the air bubble gets into your blood, it's basically fatal. Basically, just blood is removed. You have a blood pump. There's a hairpin which prevents. I'm sorry, heparin. I keep saying hairpin. Heparin is added to prevent blood clotting. And then you go into the actual dialyser itself, which has it's basically a partially permeable membrane between the blood and actual dialysis fluid. Um, you need to roughly know the two forms of dialysis. So yeah, once you're in the actual dialysis bit, there's a partially permeable membrane between your blood fluid and a dialysis mixture, a dialysis fluid. The fluid contains the correct stuff and the blood contains bad stuff. So anything which you have too high concentration of will go into the dialysis fluid. Anything you have too low a concentration of will move from the dialysis fluid into your blood, and blood then goes through an air trap to remove air bubbles and back into your body. So that's the essential of hemodialysis. In peritoneal dialysis, you basically have that exact same dialysis machine, a partially permeable membrane, blood moves through it, it's placed in your body, so it's kind of happening inside your body. So one happens outside your body and goes through a whole kind of system, and one happens inside your body. The important bit to know is you have the partially permeable membrane, which if stuff is lacking, stuff will be added to it, and if stuff is too much, will be removed. It will be removed from an artery. No, will it? That is actually an excellent question. See, I would say personally, it would be a vein. Well, it is most likely to be a vein purely because okay, I don't know if it's an artery or a vein. What I do know is that it will have to be after the kidneys, because hemodialysis is to kind of clean the blood, which the kidneys would normally do. Kidneys don't work, so it can't do that. So what it's going to do, once the blood has gone through the kidneys, it will then clean it once it's gone through the kidneys, because you don't want it doing it kind of before it's gotten to the kidneys, because otherwise the kidneys could do something wrong. So it will. All I know is that will happen sometime after the kidneys. After the kidney, so it's probably an artery coming from the kidneys and then leaves in and then is returned into a vein. That's what I assume. There's a blood pump in the dialysis. There's a blood pump, so I think pressure wouldn't be a problem. Um, where is it? Moj. I've gone through. Was it Moj? No. Where am I? Yeah, it was. I've gone through evidence for chemiosmosis in a previous one. It is really kind of you. Again, it's something I don't think really comes up that often. I will very quickly answer it though. Um, it is essentially the two big ones is that there is a pH gradient, which you'd expect in the intermembrane space of mitochondria. There's a low pH rather. It means there's lots of protons there, so that makes sense. And the other one is if you have something called a mitoplast. Let me just, yeah, mitoplast, and that's mitochondria without the outer membrane. Chemiosmosis can't really happen. It doesn't happen as much, and this shows that you need that intermembrane space for a build-up of protons. Those are the two biggest pieces of evidence, but no, I don't think you really know that. Right, now you want me to do mitochondria. Very similar to a chloroplast, but right. Here's a mitochondria. And then I'm sure it's on one. Right. Just as we did earlier, right. This here is where oxidative phosphorylation occurs. So we have outer membrane, inner membrane, intermembrane space, envelope. This is a crista, plural cristae, and this is the matrix. So we have in the matrix, you have all the enzymes needed for the Krebs and link reaction. You have coenzyme NAD. You have basically all the things you need. You have mitochondrial DNA, so you can create proteins and enzymes you need, and mitochondrial ribosomes. So we'll have, perhaps a loop of DNA here with a ribosome attached to it, so it can make its own enzymes such needed for the Krebs and link reaction. Thank you. In the inner membrane, you have yet again, it can be partially permeable to certain things depending on lipid concentration and composition. It's folded into many crystals. The classic answer: it provides a large surface area to volume ratio, and it has many electron carriers and ATP synthase enzymes, so it can do oxidative phosphorylation. Those are the stalk particles are also known as the ATP synthase, and these are the carrier proteins. So basically, it contains all the enzymes needed in the matrix. You have DNA, which can code for proteins, and the ribosomes, which can make it, those proteins in particular enzymes. You have many cristae, you have many cristae, which provide a large surface area to volume ratio, and these cristae have electron carriers and ATP synthase molecule to allow for ATP synthesis. So I hope that answers your question. There's no right picture apart from this that's a very thin bit, and that bit kind of got a bit wonky. But apart from that, this half here is pretty good. As I said, I did light dependent stage in a previous video, previous live stream. So I can't remember which one that was, but I think it might have been the first A2 live stream. So if you can you watch that, purely because light dependent stage takes forever to go through. It's quite a long stage, if that is okay. If you do desperately want me to go through it, I will, but only if it's a desperate need. Okay, sure. From photosystem 1, let me just. Okay, so photosystem 1. Two E minus, which will then, I'm going to do it slightly differently from the book, just to let you know. So we have from the photolysis of water, 1 second, you have 2H plus, you have 2E minus, 2H plus, which form 2H. Oh, no, it should be a P, sorry, NADP. NADP combines with with 2H to form or reduced NADP, and that is involving the enzyme NADP reductase. Right. Okay, I hope that literally makes sense. So first we want 2 electrons combined with the 2 protons from photolysis of water to produce 2 hydrogen, which combine with NADP and NADP reductase to form reduced NADP. Right. Can I do transmission of... do you actually... Wait. Do you just mean the difference between the transmission between myelinated and non-myelinated? Because if so, I can tell you that's not going to come up in the exam because that came up in the previous exam. Oh no 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 let's go no. Sorry. No, we're in photosynthesis now. These come from the photolysis of water, where you have H2O splits to. Ooh, that's going to be a plus. Photolysis of water. That's where the protons come from. Yeah, yeah, I know it does happen, but no, the H pluses are will then be used up in chemiosmosis as well. Actually, no, right. Um, Honestly, actually, I will admit I was mistaken. Right, sorry. I, I am technically right as well. We are both technically right. What happens is the protons from the photolysis of water are the ones involved in chemiosmosis and then are used here. So yeah, so the protons go and then are used in chemiosmosis and then are used here. So we are both right. Yeah. So ignore that. It kind of does a journey like this. ATP synthase. We say this is here. Most scribbly writing ever. But yeah, so the protons move to ATP synthase, go through chemiosmosis, and then go and reduce NADP. Why is water photolysed? Uh, uh, okay, I'm going to say I do not have enough time to answer all the questions that have just been asked. Right. Why is water? Why is it photolysed? It just is. It's just in the presence of light, gaining light energy. It's going to just, I'm guessing, gain energy and split. Just like if you heat something, it can gain energy and split. It's just another form of energy. So I assume that's what it is. Don't worry, you don't need to know why though. Yes, they do. I don't have enough time to answer either the question that I've just been asked. Both Ellie and students, if you need help with that question, either watch the video on for students the kidney and Ellie on nervous system, or email me on my email, which is found in the video descriptions, and I will go through it with you personally because I've just over 3 minutes. I don't have enough time. Definitely not to go through both. And I don't, not to properly and well detailedly go through both of them. Sorry. Yes, you do need to know those compounds. Can I very quickly? I will do my best. Even though I think that's a little bit too much, but I will have a. Actually, no, I really, because I need to leave very promptly at 1:30. Um, Again, if you want to go through limiting factors, email me. It's easier. What grades I get for GCSE? I got 5 A stars and 6 A's. Um, um, all right. Well, time is running out. So I hope this helped everyone. An hour and a half does go quite quickly, actually. Are there any questions I couldn't answer or you want more help on, please email me, munroto7@yahoo.co.uk, and I will go through it with you or watch the video concerned. All my videos up until, apart from the final module of A2 biology, have been done. That includes AS, and so Unit 1, 2, and 4 completely done, and most of Unit 5. So yeah, watch them if you need help. Email me if you need help, and um, yeah, okay, you got 14 A stars. Well done. My hobbies, very quickly. Um, I'm very big on music. I play violin in school orchestra and just play it for fun. I sing in the school choir. I do musicals. I act, but it is just for fun, nothing particularly serious, mainly in school stuff. In terms of just general kind of interest and hobbies, since I'm nerdy, I'm quite into stuff like evolution, actually researching in prehistory and like dinosaurs and things like that. Yeah, right. And just let you know, in terms of future live streams, I will be doing. Um, you always love my shirts. Um, Over the half term between, well, between Easter, this half term, the next half term, that half term, the half term before our exams, I will do 2 live streams. One on Unit 5, once because I will have fully learnt it, and then one on just general A2 biology. I'll do 2 of them during that half term. I'll do more details much nearer the time. And over the next 3 weeks, I aim to get the rest of A2 biology done. And for anyone doing chemistry, I finished Unit 5 today. Unit 5 chemistry has been done. Unit 4, I've got 4 videos to do, and I aim to get them done in the next few weeks. I'll have them done by half term. I admit I'm generally quite weird in my music tastes. I'm really into big and old and jazz music and swing, so like Frank Sinatra and Dean Martin. I'm quite weird like that. But if anyone is into jazz, then they're a good person. Uh, but yeah, so thank you all. It's been a pleasure as always to do these, and you seem to have all enjoyed them. So please feel free to just drop me a message anytime you want help with anything or to ask any weird questions. And yeah, I will see you sometime. Thank you and goodbye.